hello, Alexis Komor. Thank you for joining us today on the Regen podcast for UC San Diego Sanford Stem Cell Institute. It's really great to have you here.
Well, thank you so much for having me. I'm excited to have a chat.
Absolutely. Tell us about your position here at UC San Diego and what you do within your center.
Yeah, so my primary appointment here at UCSD is in the Department of Chemistry and Biochemistry. So I started in 2017 as an assistant professor, and then I recently was tenured and promoted to associate professor.
Congratulations on that.
Thank you. And just before that, I took on a role at the Sanford Stem Cell Institute. And so I'm the deputy director of the Innovation Center along with Jean Yo. Mm-hmm. And so for the Innovation Center, we just try to accelerate the development of like laboratory science to the bedside. So trying to accelerate innovation and therapeutics so that it gets to the clinic and is helping patients faster.
Wonderful. So it sounds like you're a real integral part because it's not just, oh, we have this therapeutic and suddenly it just magically appears for a clinical trial or for someone to be treated with in the clinic. You're doing a lot of that work.
We are, yeah. So it's, inspired by, our own research, but the point is to help researchers across campus. Mm-hmm. And so hopefully it's therapeutic agnostic. But yeah, my research specifically focuses on genetic disorders and developing ways to treat and eventually actually cure genetic disorders.
That's great. How did you choose this line of work? How did you get to the area where you're really honing in on genetic disorders?
Yeah, it was a very roundabout way. And I always say this, nothing was done on purpose. Uh-huh. Everything just sort of happened by chance.
We call that serendipity in this room. Yes.
I just sort of followed the path of least resistance, and here I am. But so my training background is all in chemistry. So as an undergrad, I studied chemistry. My PhD was in chemistry, but I focused on, we would make these small molecules that could bind to DNA. And so I got into the field of DNA and genetics. And then for my postdoc studies, I really wanted to branch out a bit. So I went to work for David Liu, who was at Harvard. And I decided to go into a new field in protein engineering and directed evolution. But I wanted to keep working with DNA. And so I started working with proteins that could bind to DNA. And at the time, CRISPR was really big, which is this technology that allows us to go, into the nucleus of a live cell and change the sequence of the DNA. And so that's kind of how I ended up in this field where we're modifying the sequence of DNA, and potentially that can help to cure genetic disorders, which are caused by mutations in our DNA.
That was going to be my next question, so you beat me to it. So CRISPR is the ability to basically alter the DNA.
Yes. Yeah. So each of us have genomic DNA. It's made up of 4 letters, A, C, G, and T, strung along in a particular sequence. And so mine is different from yours, and that's why we're all unique. So many of these differences in our DNA, which we also call genetic variation, are, not bad for our health. They're good. They make us unique. But sometimes you can have a mutation that causes something like sickle cell disease. So that would be something that has a negative impact on your health. And so, CRISPR is this large protein. So this like macromolecule, we call it. And it binds to a piece of RNA and then goes to a specific location in the genome, like a homing device. And we tell it exactly where in the genome to go, which is really remarkable because I mentioned those 4 different bases or letters that make up our genome. There's actually 3 billion of those all strung along. And so, out of 3 billion bases, we can tell CRISPR exactly where to go. And so it goes there and it binds. And then from there, that's kind of where my research comes in, is how do we then kind of change the sequence of the DNA at that spot in a particular manner of our choosing.
That is, it sounds to me like cutting edge science.
It is. It's like science
Right.
fiction.
Right,
But
exactly.
it's nonfiction
It's
now.
like something that we'd see in a movie from, like, we've seen this movie before. We're
Jurassic
altering-
Park.
Right. Thanks. Yeah. Yeah. Let's give them a plug. Yeah. No, that's great though. That's pretty amazing to work on that. What did you ever think that you'd be at this point in your career working on something like this? Like take it back. How did you even think about, this is the pivot, this is the direction, whether it's through serendipity or luck or whatever?
Yeah. Again, everything was just kind of following the path of least resistance. So, CRISPR was discovered, not so much, it had been discovered for a while, but it wasn't really understood what it did or how it worked. And so that was figured out in 2012 by Jennifer Doudna and Emmanuel Charpentier. That was sort of the big defining thing that got them the Nobel Prize. They figured out what it did and how it worked.
And what year was that? Do you know?
That was 2012.
Okay.
When their big paper came out. And then in 2013, a couple different groups were able to get that sort of molecular machine and use it in live human cells in a dish to do genome editing, which is the word for changing the sequence of the DNA.
Mm-hmm.
And so the way that it works, so I told you it's like a homing device. You tell it exactly where in the genome to go and bind. The original CRISPR system then cuts the DNA. So sort of like molecular scissors is what we call it. It goes and it cuts it. And that ends up sort of disrupting or kind of changing the DNA sequence, not really in a controllable way. So it'll like cut out a base or two and then put the DNA back together. So it's some people call it genome vandalism,
Mm-hmm.
but it actually is a super, super powerful, useful technology. So that's what was used in this FDA-approved CRISPR medicine, Casgevy, that was developed by Vertex. They used just, regular CRISPR 1.0 molecular scissors going in and cutting the DNA and kind of disrupting the sequence there. So that was the original technology.
Okay.
And then when I kind of started my postdoc and got into the field, and I was this, very unsuspecting DNA chemist coming in, deciding to do some biology, my postdoc advisor said, so there's this really, powerful, useful tool, CRISPR. We can go anywhere in the genome that we want. The problem is we don't want to cut the DNA. We don't want it to be molecular scissors. We want it to be more like a pencil and an eraser. And so that was when I developed what some people call CRISPR 2.0. And so I take Cas9 or CRISPR, I won't call it Cas9, it's a specific type of CRISPR. So we take CRISPR, bring it to where we want in the genome. And then what I do is I kind of attach it to another protein that does a chemical reaction. So this is kind of where my background in DNA chemistry came in. So I developed these tools that could do chemical reactions on the actual DNA bases. And so, as I mentioned, there's, A, C, G, and T.
Mm-hmm.
So I developed two different ways to do chemistry to these bases. In one way, we chemically convert C to T, and then in the other one, we chemically convert A to G.
Wow.
And so we can install what's called point mutations.
Mm-hmm.
So instead of a C having a T, that's called a point mutation. And so now we can really be very precise and control exactly what changes we make to the genome.
That is, I don't have any other better word than cool. Like that is just, that's amazing to me that even happens.
It was amazing to me that it happened too.
Yeah.
That the idea actually worked.
And that's the type of work that you're doing here at the Sanford Stem Cell Institute.
Yeah.
And the Innovation Center.
Exactly.
That is great. Well, that would make sense then why Endpoint News recently named you one of their 20 under 40 for the next generation of biotech leaders. How's that feel?
I hope that I can live up to that. So I think I'm on that list because the technology, the CRISPR 2.0 that I described, it's called base editing. And so that IP or the patent for that technology was the basis for two companies. So Beam Therapeutics, which develops base editing for human therapies against genetic diseases. And then also Pairwise Plants, which they license the IP for use in plants and for agriculture applications. And so they're doing some really cool, it's not as flashy as human therapeutics, you know, but I mean, the stuff that they're doing is awesome. They're like trying to make blackberries with no seeds, cherries with no seeds.
Wow.
They made this new like salad lettuce. I digress. But so, and then there have been a couple other bigger companies that have sub-licensed the tech for certain applications, such as Verve Therapeutics. So they're using base editors to treat different like heart diseases and high cholesterol and whatnot.
Well, that would make sense that if you've got the background in all of this, why Endpoint News called you unstoppable and credited you with engineering CRISPR 2.0. So what's CRISPR 2.0?
Yeah, so that's the base editing technology.
Okay,
so
okay,
CRISPR 1.0 is the scissors.
Yes.
And then CRISPR 2.0 is now like a pencil and an eraser.
That makes sense
where,
to me.
Yeah, we can change one base to another very precisely.
Wonderful. Thanks for making that connection. You just didn't call it CRISPR 2.0, but you already explained it. That's awesome. So you had technology that you developed, and it was recently used to develop a bespoke gene editing therapy that appears to have at least partially reversed a deadly rare disorder in an infant. Tell me about that.
This was crazy. I was not involved in the study. But they used the technology that I developed, so base editing, and so there are two main researchers at UPenn, and this baby was born with what's called a urea cycle disorder, and so the body basically can't properly process protein and different things that are in our diet. And so they get this huge buildup of ammonia, and it's very deadly. And so a lot of these babies actually don't live past 6 months. And it's like very obvious in the first, weeks of life. They knew something was wrong.
Yeah.
And so they did genome sequencing to try to figure out if they could pinpoint what was wrong. And so they found 2 mutations in this gene that's very important for breaking down protein. And they were G to A mutations. And so that one CRISPR 2.0 base editor can do A to G mutations. So it could actually revert the mutation back to the normal sequence.
Which explains the pencil and eraser theory as opposed to the scissor theory.
Exactly.
Yeah.
The thing is, though, I mean, this was a very unique mutation. So nobody had ever really seen it before. Nobody had ever tried to correct it before. So with some diseases like sickle cell disease, there are lots of people that have it. And so tons of research has been going on, like focusing on this particular mutation and trying to correct it. This was, very rare. Nobody had seen it. And so it was, they had to start from square one to try to figure out how to come up with a strategy. They knew, okay, we're going to use base editors in general, but there's, a lot of details about how to go about like which type of base editor. I mentioned that CRISPR uses this piece of RNA to do editing. So we had to figure out what sequence of RNA to use. And the clock was ticking, like literally this baby had, not that long to survive. And so it was a huge tour de force. They did all of this work to both figure out how to correct the mutation, as well as get a full safety profile for the FDA so that they could say, okay, it's safe enough to administer this new, therapeutic to an infant. And so they were able to, I think, dose the baby at maybe 7 months. And he just had his 1-year birthday. He got to leave the hospital. His name is KJ. There was a New York Times article about him. It was just really amazing. Everybody is like so happy that they were able to get this done so quickly. And I mean, I really want to focus on, the safety aspect. That's not, really what I do. I kind of more develop the technology on the front end. But, they really were very rigorous. They didn't cut any corners. They did their due diligence, and it paid off.
Yep. And they legitimately used your technology to influence and extend the life of an infant who otherwise may not have had that opportunity.
Right.
Thank you for that. Thank you for what you do.
I mean, it wasn't just me. It was, all these clinicians, everyone all working together. Yeah. A village.
Yeah. Takes a village, they say. Absolutely. Polygenic disorders. I would be remiss if we didn't ask you about them. First of all, what is a polygenic disorder? And then how does the work that you've done play into potential therapeutic treatments for those?
Yeah, so this is I wouldn't say my specialty, but my what I really am super into right now. And so sickle cell disease is what we call a monogenic disorder. There is like a single mutation in one particular gene, and that's it. That's all that's required to cause this really awful disease. Most diseases, though, they aren't caused by just a single mutation somewhere in the genome. It's lots of mutations across many different genes that all sort of collectively have to be there in a specific combination to cause the disease. And it's really hard to do genome editing at just one site on the genome. So when you start to think about, I think like diabetes, heart disease, if you look at the risk factors for these, I mean, there's like 20, 30 different genes and point mutations that can all kind of collectively contribute to the disease. So thinking about like, how are we going to scale up when, doing at just one location is difficult, scaling up to 20, 30, 40,
right.
That's crazy. But I'm really, really, really into this because I think that like the vast majority of genetic disorders are not going to be monogenic. And they're going to be caused by two or more mutations. And so we recently developed this system that we called it multiplexed orthogonal base editing, which shortens to MOBI. So you always have to come up with a catchy title.
Always have to have an acronym.
And so a couple of people, they love the MOBIs. So we came up with this way to multiplex our base editors. And so we can do, as I meant, there's kind of two flavors of base editors, the C to T base editors and the A to G base editors. And you weren't able to use both in the same cell at the same time. I don't want to go into the nitty-gritty details of why, but when we developed this MOBI system, we were able to re-engineer the system to overcome that. And so now we can do multiplexed editing, and the efficiencies are really quite good.
Got it. So the therapies you're doing, they're not so myopic. It's not just one treatment for one thing. You're looking at the complexities of polygenic disorders, diseases, whatever it might be, and how to tackle that in a holistic way on a mega scale, as opposed to just treating one piece of it one time.
Right. And so one thing that we think, isn't super kind of well appreciated is the fact that if you look at human sequencing data, so we've sequenced lots of humans and we found lots of point mutations. And most of them are kind of in this, like they're called unclassified. We don't really understand whether they're pathogenic or benign, so, causing a disease or not. And when you start to look at out of all the hundreds of millions of genetic variants, and then you look at, but here are like the ones that are known to cause a genetic disease, and that's what we're kind of developing therapies against. It's like 0.1%. And so that's kind of one thing that we're trying to better understand is, why do we not understand how these variants, or whether or not they're causing disease or not? And we think it's because you have this combinatorial issue where, this mutation could be pathogenic, but it needs to be occurring along with various other ones in the genome. And it's really hard to deconvolute that out of this human sequencing data.
Yeah. It's multi-layered.
Yeah.
And you can't separate it.
Yeah. And so actually a lot of the work that we do is on the side of modeling different point mutations. So generating cellular models where we have a variant or one or two or three or four, using our Mobies to install lots of them, so that we can tell, okay, is this mutation pathogenic, or is it only pathogenic if it's occurring along with these three other ones?
That makes sense to me. Yeah, it's complicated, but it makes sense to me. So that is an amazing summary of the amazing work that you've been doing across the industry. How is some of that work now being translated into the work you're doing at the Sanford Innovation Center through the Sanford Stem Cell Institute here at UC San Diego?
Yeah, so a lot of this technology that we've built for, genetic disease modeling or correction, it's not super accessible to other labs that aren't experts in using genome editing tools. So one of the kind of pillars, so to speak, that Jean and I have built up in the Innovation Center is more automated ways for labs to come in and they say, I am an expert in X, Y, and Z disease. I know that this particular gene, mutations in it are what cause this disease, but we don't know which ones. And so we kind of help enable them to do studies where they can do genome editing in their model cell line, and so that they can start to model or correct mutations in this disease that they're an expert in, that I don't have expertise in, but I do have the expertise in on the genome editing side of things. One other thing I'll point out is, as I kind of mentioned with the baby KJ example, even if you do know this mutation here is causing this disease, we in theory know how we could correct it. There are still hundreds of different kind of combinations of editors and pieces of RNA that you could use to theoretically correct that mutation, and you just don't know which one is actually going to work. And so kind of a lot of what we do in the Innovation Center is like helping with scale up too, to kind of do experiments in high throughput. So, here's a mutation, we want to correct it, but there are 1,000 different ways that we could. So we try to help people kind of scale that up into like one pot, so to speak, where, okay, here are those 1,000 different ways that you could, in theory, correct this mutation. We're going to do it all in one experiment and figure out which one is the best way for you.
And that's where the modeling comes in, to be able to develop that model so that it's more of a turnkey process. Yeah.
Okay. But yeah, everything is very much specific to whatever disease that they're working with, and whatever cells are specific to that disease. It's all very, very customized, but that's kind of like what we're trying to help with, is like customizing these things, but still kind of accelerating them forward.
Yeah. You are so enthusiastic about your work. What keeps you motivated to do this stuff?
I mean, this is a lot of scientists. We're not motivated by the money, certainly. It's all about kind of understanding the unknown. And what keeps me up and would this work? Would this work? Kind of like I'm always kind of thinking 1,000 miles a minute about here's a problem in the field. How do we tackle it? I have all these ideas. I certainly don't have time to go in the lab and do it, but I have, graduate students and postdocs who are just excited as me, and I'll come in and I'll say, oh, I came up with this really cool idea, but I don't know if it could work, and we'll work through it on the whiteboard and draw an idea, and then they'll go, oh, well, this part won't work, but maybe if we do this instead. And it's sort of like a puzzle, and you're kind of putting all the pieces together, and then maybe you kind of put this region together incorrectly, so you kind of undo it and someone else helps you put it in. So I love puzzles, and it's all sort of discovering new things and trying to thinking about big problems and figuring out how to get there and how to combat it.
It sounds like regenerative medicine ended up being your sweet spot then.
Yeah, definitely. So again, nothing that I did was on purpose. I just, went with the flow. And I, originally was, again, a chemist. I was doing synthetic chemistry, mixing things in round bottom flasks and Erlenmeyers and whatnot. And I kind of ended up in this field accidentally, but then when I was there, I was like, this is amazing. This is what I want to do. And that's kind of how that you've kind of hit your niche is you'll know. I talked to a lot of young people, students, undergrads, grad students, and they said, well, how do you know when what you want to do when you grow up, so to speak? And I tell them you'll know. If you don't know yet, it's because you haven't found it yet. But just keep going, and eventually you'll get there, and you'll know this is what I want to do, and I can't imagine myself studying anything else.
That's really great advice for the next generation of folks that are coming up that will soon be your colleagues, and then eventually take over the work that you're doing years from now.
Yes,
right?
when I'm ready to retire.
Right. Don't retire yet. You're not allowed to go anywhere yet.
I know.
Let's talk specifically about one more example from your research. Your base editing method was also used to help create a treatment for a young girl with leukemia that required genetic editing at 3 separate sites. And that was something that I guess CRISPR couldn't accomplish. So talk to us about that.
Yes. So again, polygenic disorder. This is not a polygenic disorder, but it's the same kind of idea where you need to do genome editing at multiple sites in the genome at the same time. This is why I say, multiplexing. That's my thing. So this particular example, this was actually the first example of base editing being used in a person. And so this patient had leukemia, and the PI who led the work, he's a leader in what's called CAR T cell therapy. And for our listeners, what is CAR T cell therapy? So it's a fancy word for you kind of take immune cells out of a healthy person, and then you tinker around with them and make them able to recognize cancer cells and then kill them. And so it's been a huge revolution in the cancer field. It's very effective against blood cancer specifically. But since this was a little girl, she had leukemia, and then they were engineering these CAR T cells, or these cancer killing cells, out of a different person. Those cells are not always compatible. So if you put them in her, they're going to attack her healthy cells too, and some other issues with compatibility. And so to make them compatible, you have to do additional engineering where you kind of cut out certain genes that are important in certain circumstances, but all we need these cells to do is to kill cancer, so we don't need them. And so when you try to do that with CRISPR 1.0, which is usually very good at disrupting genes and getting rid of them, if you're trying to do it at multiple sites in the genome, you're cutting your genome up in like multiple little pieces, and they can come together in the wrong order.
Okay. All right.
And so that was an issue with generating these CAR T cell therapies previously, was how do we kind of disrupt these multiple genes? So in this case, it was 3 specifically. When we're getting, the genome is coming together in random orders and kind of being, that's really not good. But so what they could do then is they used base editing, and they looked through the 3 genes that they wanted to turn off or get rid of, and they were able to identify C to T mutations that would turn off the gene and get rid of it. And so that's how they were able to knock out or turn off or disrupt, get rid of those 3 genes all at once with one genome editing strategy, was using the base editor.
Okay.
So again, multiplexing is the best.
Absolutely.
I love multiplexing.
Yeah. And I have to say that, in addition to your enthusiasm, you do such a great job of synthesizing these really, really difficult scientific theories into digestible, tangible tidbits for folks to understand. So with that vein, why is science important? Why should people care about science?
Oh, man. Everything, all of our everyday items, I guarantee you, like some aspect of what, like our cell phones, our computers, maybe you take a drug for some chronic disease that you have. I guarantee you, like all of those things, there's some aspect of it that originated in an academic lab. Because again, in academia, we're doing really basic research. We're doing the totally crazy stuff that companies are like, I'm not going to touch that. Like, we don't know if that's going to work. But yeah, we're able to try it. And, a lot of times it pays off. So this base editing idea was absolutely nuts. The only reason I even tried it was because I was this naive chemist trying to do biology, and I was like, of course this could work. Why not? And everybody around me was looking at me like, you're nuts. But again, these ideas, they pay off.
They do.
And also, thank you so much for saying that I'm able to kind of break these terms down. It's because I tell my parents about my research all the time. they're really interested, but they do not have science backgrounds.
That's fair.
And so I practice on them.
Yeah.
But yeah, no, what we do is so important. Again, it's especially in academia, it's the crazy stuff that is super high risk, high reward. And so, the companies they're too risk averse to try in most cases. And, it's just so important.
Yeah. It's amazing that and you've said it a couple of times, that these are the crazy things and the crazy ideas. But I think UC San Diego and other academic institutions provide a unique baseline and foundation, founded in science, founded in research, but also founded in academia, that be able to provide some of those guardrails for that crazy research that ultimately, at least in your area, has resulted in very tangible therapeutics that make a difference in people's lives. So it's not so crazy.
Yeah, so I shouldn't say, oh, we're totally nuts. This is, we're doing all of this research on a lark. Right. When we write grants, we have like half of the grant is a list of like, this is why I think it's going to work, even though it's a crazy idea.
Yeah.
So we've done our due diligence. And also, one thing I just want to plug UCSD. The environment here is so supportive, so collaborative. Like, I have never seen, a place that's this supportive and this collaborative. Where, again, I'm in a chemistry department, and I'm constantly collaborating with people in the med school, people in pediatrics, all over campus. And that really, bolsters our science too. When I say, we write these grants, and we have this long list of reasons why we think it's going to work. The reason we came up with those reasons why we think it's going to work is because I, took my colleague to lunch, and I said, I have this crazy idea. Let's see how we can make it to work. So it's just the collaborative and supportive environment here is outstanding, and it's the best place to do science.
It is collaborative. It requires multidisciplinary and interdisciplinary research. And I appreciate that at least we, the Sanford Stem Cell Institute, gets to be at a special place like UC San Diego that isn't so myopically thinking or so siloed that folks don't want to work with each other. And so I think that lends itself well to your philosophy on science and working across the aisle, so to speak, with other folks from other departments that might not otherwise work together.
Yeah, and that's exactly what we're aiming to do with the Innovation Center. Is anybody who has some of these crazy ideas that eventually will become a therapeutic, and come and talk with us and talk with your colleagues, and we're here to really accelerate that forward and make it work.
That's fantastic. What's next on the horizon, without giving away any secret sauce or something? What are you working on, or what are you hoping for is the next innovation, or even the next step that you're building on an innovation that's already there? What's coming down the pike?
So we talked a little bit about developing new ways to edit the genome and going after new diseases and understanding the causes of diseases. But I think one thing I haven't talked about is what we in the field, we call it the delivery problem. And so that's the fact that, so let's take, baby KJ. He had this, urea cycle disorder. And basically the way the cells in the body that needed to be corrected were liver cells. So that's actually like a low barrier, where all we had to do was deliver the editor to the liver cells in order to correct it and make him better. But think about, but what if we had, a mutation that causes Alzheimer's, and we want to correct that? That would mean that we would, we need to do genome editing in the brain. So again, how do we deliver these editing components to the brain, to the heart, to the lungs, et cetera? So I think that is currently the huge bottleneck for a lot of technologies, is just generally delivery. And so again, shameless plug for the Innovation Center, but we also have a setup to generate these adeno-associated viruses, or AAVs, which are currently being developed for ways to deliver not just genome editors, but lots of different therapeutics that are based on protein components to different areas within the body.
Yeah, that's pretty great. It's pretty awesome. Yeah, I grew up in the 80s, so I get to say awesome a lot. Alexis, you've been amazing. Thank you for the conversation, and thank you for the insight into the work that you do. Congratulations on the accolades and the coverage and the awards that you've been getting. I am esteemed to call you a colleague. I love working in the Sanford Stem Cell Institute with you. So I would definitely want to have you back on the podcast. And thanks for your time today.
Thank you so much. This was a lot of fun. I'm happy to come and chat, and I can bring my parents next time too.
We could
whatever
do
you
that.
want.
I like that idea. Thank you so much.
All right.
All right.